Okay, we'll get going for tonight. So I'd just like to welcome everyone back to our fourth and final instalment of the MND Research Australia State of Play Research Seminars. First of all, I'd just like to perform an acknowledgement of country. So in the spirit of reconciliation, MND Australia acknowledges the traditional custodians of country throughout Australia and their connections to land, sea and community. We pay our respects to the elders past and present and extend that respect to all Aboriginal and Torres Strait Islander peoples today. And I'd like to acknowledge the Wiradjuri people on whose land I am and pay my respects to elders past and present. So for those who haven't joined us previously, these sessions are replacing our usual MND Connect session, which we have live and in person. So instead, we're bringing the research right into your living rooms and offices instead. Previously, we've heard about how we can better model disease in the laboratory, how we can improve care models, and also how we can better measure disease. Tonight, we're going to hear from Dr. Derek Stein and Dr. Richard Gordon, both from University of Queensland. And they're going to tell us about new pathways to treatment or new ways we can possibly approach developing new treatment modalities. So Derek's going to tell us about his work in looking at the metabolic system in MND, and Richard's going to talk about his work looking at inflammation in MND. So during the talks, please feel free to ask questions, either using the Q&A button, the Q&A or the chat function within Zoom, or if you're on Facebook, you can ask questions through the chat function there. Just keep firing in your questions throughout the talks, and we'll have a joint Q&A session at the end of the session. So Derek will talk first, then Richard, and then we'll have the Q&A section. So without any further ado, I'd like to hand you over to Derek, who will take us away first. Thank you, Derek.
Okay, so thank you very much, Jason. Can everyone hear me okay?
Yep, all good. Yes, all good.
Perfect. Thank you very much for this opportunity to speak today. I've given this talk to a lot of audiences before. This is the very first time I'm actually presenting this data online. So we'll see how it goes. Lots of questions, I hope. I've been asked to talk a little bit about the work we're doing in metabolism, and this is a very broad topic. And so what I'm going to do today is really just give you an insight of what we actually mean when we talk about metabolism. I'm going to discuss this idea called hypermetabolism, which is observed in people with MND, and talk a little bit about why this might be important. And you'll see throughout the talk that really the theme is what we know, what we do not know, and what we don't know, and what we actually need to know. There's a lot of questions that we still have to answer. But before I do that, I thought I would just take a little bit of a step back and as a priority, talk about why we do this research. So for those of you who have joined us over the last few weeks, you would have heard this. When we talk about MND, we talk about a group of diseases under the term MND. And these diseases all have one thing in common, and that is the fact that people with MND tend to see the loss or the death of their neurons either in the brain, these are the upper motor neurons, or in their spinal cords, and these are the lower motor neurons. And depending on how these neurons die, people will be placed in these categories of different types of MND. So as an example, people with primary muscle atrophy will experience predominantly loss of the neurons in their spinal cord. So these are the neurons that send long projections out to the muscle and attach to the muscle and tell the muscles what to do. And when these neurons die, they pull away from the muscle loses connection with the brain and the spinal cord. And as a consequence, people experience lots of muscle wasting. On the other side of the spectrum are people with PLS, or primary lateral sclerosis, and these individuals will experience predominantly the loss of these upper motor neurons, the motor neurons within the area of the brain that controls movement. And of course, when these neurons die, these long projections into the spinal cord, they disappear, which means that the motor neurons in the spinal cord no longer receive information from the brain. And as a consequence, while the muscle still stays full, people progressively lose the ability to use those muscles. And over time, they'll experience much faster reflexes because the brain is not controlling the reflex response. And these individuals also, like people with PMA, experience a high degree of disability. And of course, there's this very common form of MND called ALS. And really it's ALS that's that combination of both upper motor neurons and lower motor neurons. And this can start in the arms, or it could even start in the throat, or the area of the body that controls swallowing and speaking. And that's called bulbar onset. Now, it's very important to recognize these different types of MND because, of course, depending on what diagnosis you have, you'll also have a very different course of disease. And unfortunately, everybody with a diagnosis of MND will pass away or die from MND. However, the change, I should say that the course of disease and the survival of MND across these different subtypes of MND is quite different. Some people passing away within months to years of diagnosis, whereas people with these more upper or low motor neuron dominant types of MND can have a much, much longer survival, 10, 25 years or more. Now, one of the things that we don't always focus on is the fact that even within the different subtypes of MND, we tend to see huge range, huge variability in survival. And so here I've just pulled an example of a review that was published earlier this year, looking at the variability in survival with people with MND. And the authors did a really wonderful job in pulling together a range of publications dating back all the way to the late 70s. So there's about 40 papers that they pulled together in this particular table, and I've just put it into some pictures so we can see what it looks like. And these are all studies specifically in people with a diagnosis of ALS. And you can see here that the mean survival of those individuals is quite different across those different studies. And while there was on average about a 40-month survival following the onset of symptoms, you can see that in some instances, the mean survival of that cohort was all the way up to 120 months almost. And what's really important if we look at those individual studies is that even within those studies, there's a huge range of variability. And so here's just some examples. And you can see that, between 12 to 28% of some of these studies, 12 to 28% of some of the individuals in these studies survive for 10 years or more. Now, this is very important because it means that something unique to these individuals, allowing them to either tolerate the effects of MND or something makes them unique so that the MND doesn't progress at that same rate. And really the idea is that if we know what's going on with these individuals, we can use that information and develop therapies or treatments, or even just interventions in clinical care that's going to help us delay the progression of MND for pretty much everyone else with MND. And so there's a lot of really important information within this. And later on, Richard's going to talk a little bit about the notion of inflammation and how inflammation might have a big impact on disease progression. Certainly in this review, they talk about the improvements in care and the multidisciplinary clinic and how that has had a big impact on survival. And in fact, if you were to get a diagnosis today with MND, you will have better survival than what you would have had if you had been diagnosed 25 or so years ago. Now, the other association that these authors highlighted was this association between weight loss and survival in MND. And this is something that has been observed for a number of years now. And so for those who are not familiar with this association, generally speaking, people who lose weight throughout the course of disease and people who lose more weight throughout the course of disease tend to have a more rapid progressing disease and sadly a shorter course of disease. And what's really interesting about this observation is that it's even true if you were to look at people prior to their diagnosis. So in this particular paper that was published earlier this year, the authors looked at 2,420 patients with ALS, and they found that those individuals prior to diagnosis who lost more weight tended to have a much more rapid course of disease and shorter survival. And so clearly within this idea that weight loss itself can impact the progression of disease has emerged this notion that we can actually intervene, slow weight loss, and in doing so, we can slow the progression of MND. Now, the reason I focus on this paper is because I thought that the authors presented a very eloquent description of all the different factors that might contribute to weight loss. And I don't think that they covered everything, but they certainly made the point that weight loss and MND can occur due to a number of reasons, including the fact that MND does affect the muscle. And so when people get MND, their muscle mass will naturally decline. But the authors also talked about one aspect that I think is quite important to consider, and that is the metabolism of the individual that has been diagnosed with MND. Now, metabolism is actually a very big word because it actually covers a massive range of research. And so for the purpose of today, I'm just going to talk about one aspect of metabolism, and that is the notion of body weight regulation. So we all know that when we eat food, we gain weight if we eat too much, and if we don't eat enough, we lose weight. And that's because when we eat food, we're trying to match the amount of energy that our body is burning and that we expend. Now, when we look at our metabolism and the amount of energy we expend, we can actually break it up into different categories. And for the purpose of today, I've really just highlighted 3 of those categories and roughly the percentage of energy we spend. And you'll see that we spend about 10% of our energy just to break down the food and digest and absorb it. And depending on the individual, maybe 20% of our energy goes into our activity. But really, the majority of the energy we spend every day is this thing called our resting energy expenditure. And so this is the energy that we spend every single day just by being awake and being alive. It's the energy that goes into running our organs, keeping our brain active, keeping our muscles active, and keeping our organs doing their job. And generally speaking, depending, of course, on the activity of the individual, that is by far the biggest component of energy that we spend every day. Now, the important thing, of course, is that when we look at MND, that a lot of people actually experience this thing called hypermetabolism. And hypermetabolism, very simply, is the notion that a person is burning more energy at rest than they should. And of course, the idea, if you look at the literature, is that this hypermetabolism causes an increase in a person's energy expenditure, which in turn is going to compromise a person's ability to maintain their energy homeostasis. And if you burn more energy than you consume, the result is weight loss. And so it's been proposed that hypermetabolism is actually driving a lot of the weight loss that we see in MND, and by proxy, this weight loss is then associated with a more rapid progressing disease. And so when we first started this work, our team had a look at all the literature that was published in the 25 years preceding our studies. And we found that there was huge variability in the studies and how people conducted these studies and what they reported. And so this line here just indicates when we first started showing a serious interest in looking at patient studies in the context of metabolism. Now, the other thing that we saw in these papers was that nobody really addressed the notion of hypermetabolism causing weight loss. And while they suggested it, it was never tested. But more critically, none of these papers really looked at the clinical consequences of hypermetabolism. While they reported hypermetabolism in people, they didn't really tell us whether or not this was associated with some sort of outcome for the patient. Is it associated with more rapid progression or earlier death, or anything that tells it that this is clinically relevant. And so we initiated studies in 2005 called the MND studies. And these studies really did focused on the prospective assessment of cases and controls, looking at changes in their resting energy expenditure throughout the course of their disease, or just at baseline for those controls. And the idea was that people would come back routinely every 3 months for as long as they can, so that we can just monitor how their bodies are burning energy, and specifically in the context of MND. Now, this study is still going strong. We're in our 5th year now, and I'm happy to say that in the coming year, we'll finally start looking at those longitudinal measurements. And I want to thank the 250 or so people who's already contributed to those studies. It's been an absolutely wonderful effort by everybody involved, and in particular, those people with MND, because it's not easy to keep coming back for these assessments. And for today, I'm just going to talk about the baseline results. We were able to publish a small set of data in 2018, just looking at some of the individuals that contributed to that very first research visit. Very quickly, people came in. We had measured their resting energy expenditure using a machine called the BOD-POD. Some of you here today will recognize that. And we also looked at the metabolic rate using a technique called indirect calorimetry. And I can talk about that in the questions if you want to. Very importantly, we managed to measure this information, and then we compared it to the clinical course of their disease. That includes the duration of their disease, but we also looked at things like changes in weight and composition. And our questions were, does hypermetabolism contribute to weight loss? Is it clinically relevant? Who becomes hypermetabolic? And more importantly, can we do something about this, assuming it is relevant? Now, for those of you who haven't participated in the study, this is what the room looks like. So here we have Associate Professor Robert Henderson, He's the neurologist we work with very closely. Rob is a fantastic collaborator, and he's just posing in this photo to give everybody a bit of an idea of what we do. Here we have a machine called the BOD-Pod. This is a very honest machine. It tells you exactly how much body fat you have and how much muscle you have. I personally don't get into this machine. And this is important. Well, it's important that I don't get into the machine, but it's important what it tells us because it gives us an indication of how much muscle a person has. And from that, we can we'll work out exactly, well, not exactly, but it's estimate how much energy that person should be burning throughout the day. And then after people sit inside this machine, they lie on this couch. We breathe the air that they consume and that's carbon dioxide that they produce. We measure that. And then from those measurements, we can actually work out how much energy they are using. We compare these two and we can say, well, this individual might be using more energy than is predicted. And if they are using a lot more energy, we can say, well, this individual with a relative level of certainty is hypermetabolic. Now, very quickly, I'm not going to go through all the results in a lot of detail, but one of the most important findings was, of course, confirming that within our study population, people with MND tended to have hypermetabolism. In fact, when we compare that to the control individuals in that study, we actually found that if you had MND, you were 5 times more likely to have this hypermetabolic state, this really high increase in your resting energy expenditure. Now, of course, our question was, does hypermetabolism cause weight loss? And so we actually followed these individuals for a number of years following their assessment. And I'm just presenting now the data for the first 12 months. And to our surprise, we actually found that those individuals that were hypermetabolic, this pink line here, were no more likely to lose weight compared to those individuals that weren't hypermetabolic. And so we kind of threw away this notion that hypermetabolism in people with MND causes weight loss. Now, of course, this was a really unexpected finding, and the conversations that were triggered as a consequence of this conversation led us to take a much closer look at how people are accessing their calories. And through those discussions, we actually realized that a very large portion of people with MND had loss of appetite. Now, loss of appetite means that people might not feel interested in their food, but at the same time, they may not be able to access their food in the same way. And so these are the individuals that are actually at risk of losing weight because they can't necessarily adjust their intake to meet their metabolic rate. Now, this is a story for another day. What I want to do is come back to this story on hypermetabolism because, as I promised, one of the reasons we did this study was to see if hypermetabolism itself actually is clinically relevant. Now, again, here, what we've done is just plotted the ALS FRSR scores of the people in our study. And you can see the red line here is the decline in the ALS-FR scores for the 12 months after they came in for that research visit with us. And as you can see from this graph, people that were defined as being hypermetabolic on that research visit tended to have a much sharper, much steeper decline in these functional scores, which would in this cohort suggest that those were the individuals that actually had a more rapid progressing disease. Now, during the study, during those 12 months and in the follow-up, we unfortunately had a few people pass away from MND. And so we also looked at the survival of people with MND that were hypermetabolic. And unfortunately, we found that those individuals that were hypermetabolic were also at risk for an earlier death. We found that there was double risk for death within that first year after their assessment, showing that they were hypermetabolic. And so ultimately, when we look at that very early study we've done and published in 2018, we found that hypermetabolism alone may not contribute to weight loss. We have to factor in things like changes in appetite and the capacity of people to maintain their energy intake. But we also found that hypermetabolism is quite relevant in terms of the clinical outcomes of individuals. Now, we want to change this. We want to intervene. We want to address this. And so really the next question is who becomes hypermetabolic and why? And once we've done this, can we do something about this to improve outcomes for people with MND? Now, one of the things we did within that study was we actually looked very carefully at the clinical presentation of people, and we considered whether or not these were individuals that had predominantly lower motor neuron or upper motor neuron deficits. How do they fall within that spectrum of ALS? Do they have a lot of neurons in the brain dying? Do they have a lot of neurons in the spinal cord dying? And you can do that crudely by looking at some of the clinical presentations of these individuals. For example, if somebody comes in with a lot of brisk reflexes across many of their limbs and they had increased tone and spasticity, you can score that. And you can say, well, okay, this individual tends to have a very high upper motor neuron burden. And by proxy, you can also have a look at things like weakness and muscle wasting. And again, you can score that across all their limbs. And you can say, well, this person also tends to have a very high low motor neuron burden, a type of disease that's affecting the spinal cord. And when we look at these numbers and we compare them to the metabolism of the individual, we were quite surprised to find that people who had a much higher degree of disease in their spinal cord tend to have a much higher metabolic index. In other words, these were the people more likely to be hypermetabolic. And at the same time, we had a look at whether or not people in the study developed disease, first of all, in their throat, in their bulbar region, or their spinal region. And we actually found that people who develop disease in their spinal region, these are the areas of the spinal cord that innervate those big muscle groups associated with our arms and our legs, they were also more likely to have a high metabolic index, more likely to be hypermetabolic. And so from these results, we formulated this hypothesis that maybe hypermetabolism actually occurs in the muscle as a consequence of those spinal cord motor neurons dying, pulling away from the muscle, and the muscle not behaving in the way they should. And so the next thing we did in this study was to ask those individuals in our study if they would be willing to provide a small muscle biopsy, which we can then study. And I'm very happy to say that a lot of people said yes, they're willing to do it. And we were able to actually progress a lot of this work and find some interesting observations that we published earlier this year. And so I'm not going to go through a lot of details in this particular paper. It gets very heavy very quickly, but I'm more than happy to answer any questions on this paper. It is open access, and so if anybody has questions, just email it through, and myself and my colleagues will answer it. But very quickly, what we did is we collected the muscle from these individuals that went through those same metabolic assessments, and we looked at how those muscles used energy using a machine called a seahorse. And it's a pretty cool machine because when we grow these muscles, and we put them in a dish, and we put them in this machine, we can start challenging them. And we can have a look at how these muscles in the machine use energy, what kind of energy they use, do they like fat or sugar? And more importantly, if we compare cases to controls, we can see if there is a change in how those muscles are accessing energy. And so, again, I'm not going to go through all the data, but we did find some interesting and very promising results that has changed a lot of our theories. So in this graph here, what I've done is just presented the use of fat by these muscle biopsies. And you can see that as a person's resting energy expenditure goes up, those muscles in that dish tend to prefer or actually have a greater dependency on fat as an energy substrate. So generally speaking, muscle will use sugar and fat as an energy source. Muscle loves sugar as an energy source, but it tends to look like those individuals where they have more resting energy expenditure really love fat as an energy source. And that kind of makes sense. If you have a lot of energy that you're consuming every day, your muscle is going to turn to other sources of energy that's going to use, and that includes things like fat. But this does not actually account for metabolism, the hypermetabolism of the individual. When we plot this result now alongside the metabolic index of the individual, so this group of people here are hypermetabolic, we actually found no relationship. And so this was really perplexing because it does seem that the hypermetabolic muscle doesn't seem to have an increase in dependence on substrates like fat. In fact, we actually found that's the case for sugar as well. It's not like they're trying to consume more energy. Now, I have to be very careful when I present this data because this is not conclusive. This is a correlation. And we're now working on a lot of studies to look at causation and to interrogate this in a lot more detail. But it could be that these muscles are unable to actually access the energy they need. And as a consequence, this might contribute to some of those changes that we see that result in more rapid progressing disease. And so this story is very much, well, we've published a large portion of it, incomplete, and we still haven't got the answers we're looking for. Now, I mentioned that we want to talk about who becomes hypermetabolic, and the muscle study was unable to tell us if it's people with different types of muscle and why. And this is a very difficult question to address, but we are now completing this prospective case control study, and we have generated a number of insights that we will release in the coming year once we've done some proper data analysis. And I think that the results are going to be quite surprising, because it does suggest that there is something going on within the system that we can modify. And so while those results are very, very preliminary, we have started looking at whether or not we can do something about hypermetabolism to try and improve the outcomes for people with MND. Now, I showed you the picture of how we measure metabolism in a person. Well, on this side of the screen, I actually have the system we've installed here at the University of Queenston that we use to study metabolism in the mouse. And you can see we have these two giant chambers with these red doors. And when you open those chambers, you see 8 boxes, and these boxes look like that. And inside this box, there lives a mouse, and this mouse goes about its everyday activity like nothing is happening. And in the background, we monitor it very carefully. And one of the things we can do is look at its oxygen consumption. We can look at how much oxygen it's consuming and how much carbon dioxide it's producing. So we can actually do very similar metabolic studies that we do with people in mice as well. And of course, one of the more commonly used mouse models of MND is the SOD mouse. And so, of course, we've pushed these mice through this machine to just make sure that they are an appropriate model to use when we look at metabolism. And our studies in the SOD mouse has confirmed that. And so what we've done is actually look at those mice throughout the course of MND, prior to their symptom onset, pre-symptomatic, at the time when they first developed symptoms of MND, and about halfway through them developing symptoms and us needing to euthanize those mice. And what we found was, just like patients, I should say actually just like mice who are in these chambers, these mice actually start to develop hypermetabolism as their disease progresses. Now, this is very important because it means that we can now start testing compounds and drugs in these mice and see if we can actually reverse hypermetabolism, with the idea to test whether this is going to have a beneficial outcome in terms of disease. And so I can't say much about this study because we are just waiting for ethical approval. And so this is not the name of the drug, it's just a number that we've made up. But effectively what we've done is we've given wild-type mice and SOD mice a drug that improves how tissues, muscles, and neurons access their energy. And importantly, we gave that drug to animals well after they've developed symptoms or signs of MND. And then we followed those animals throughout the course of disease to see if we can extend their survival and improve their functional outcome. But we also measured their metabolism to see whether or not this drug will have an impact on their metabolism. And the results are quite promising. So here we have animals just on a vehicle, and you can see that when we give them the drug, so here we have the animals, you can see there's hypermetabolism, a much higher oxygen consumption. We give those animals the drug, we can actually bring the metabolic rate of those animals down. And we've got some nice data that I unfortunately cannot present because it's not complete, suggesting that this drug also has beneficial effects in terms of treatment and survival. Now, we have decided to push ahead and at least do the safety studies for this compound. And my collaborator, Shuno, is just waiting for ethical results. And so hopefully next year, very early, you'll start hearing about the MedFlex study, in which we're actually going to test the safety of our compound in people with MND, with the idea that eventually we'll start doing a clinical trial to see if we can improve metabolic rate for people with MND to improve their quality of life, but also their survival. Now, that was a very, very, very quick and a very dense review of some of the studies that's been doing in MND. So I'm just going to give you a very quick summary before I pass on to the next speaker today. If you had listened to this whole talk and you needed a central message, I think it would be important to walk away to realize that MND is very different between individuals. There are different types of MND, and survival is very importantly different between individuals. And by knowing why some people progress slower, we might be able to develop therapies to help everyone with MND. One of those factors that drive changes in progression, of course, is weight loss. We know that weight loss and greater weight loss is associated with faster progression. But this is not necessarily associated with metabolism, although hypermetabolism is very common in people with MND, and that multiple factors will contribute to weight loss in MND. More importantly, it does appear that hypermetabolism is clinically relevant, and we've now started developing treatments that can target hypermetabolism with the hope that we can use these treatments to slow progression and improve quality of life for those living with MND. So I have to finish very quickly, of course, just to thank everybody across the two groups at UQ that we have the pleasure of working with. That's the STAIN lab as well as the No lab, my collaborator, Dr. Sharon No. It's a small group, are very hardworking people, and we've based all of our research studies across the clinic, but also the lab. Of course, the clinicians and the nurses that provide a massive amount of support that help us collect all of the information. I have to thank Tez, who for some reason didn't make it to the photo. I'll have a talk to him later on. And of course, our collaborators. A lot of these studies we're running across 2 sites, one in Australia, but also one in the Netherlands. And of course, a lot of the preclinical mouse studies are being replicated over in France to show that these results are trusted as well as robust. And then finally, I really have to thank the various organizations that support our research, and in particular, the MNDRA. So that's, like I said, a very whirlwind discussion on MND and metabolism with a little bit of insights into MND itself. And I spoke to Getten before, and we've decided that it'll be easier for me to introduce the next speaker, Dr. Richard Gordon. So Richard, if you wanted to maybe share your screen now and join us. So Richard is a researcher here at the University of Queensland. He's based at the Centre for Clinical Research, and Richard's work is predominantly based on the repurposing of compounds, but also Richard has a very strong focus looking at neuroinflammation and how that might be relevant to MND. So thank you very much, Richard.
Great. Thanks, Derek. Can everybody hear me okay?
I can hear you okay.
Fantastic. All right. So thanks for that amazing introduction, Derek. So I'm fortunate that we had Derek before me to give this intro, because I'm a fairly new researcher for MND. I've spent most of my life, my research life at least, investigating neuroinflammation, or how inflammation starts in the brain and how it can drive neurodegeneration. But our focus so far has been, for the vast majority of my time in the lab, has been on Parkinson's disease. So our group is fortunate to have, we recently started a drug repurposing program, and we were really fortunate to get funding from MNDRA to start our work in motor neuron disease, because there is a lot of emerging evidence suggesting that neuroinflammation or inflammation across the board, not just in the brain, but also in the gut and peripherally in MND patients, could have an important role in disease progression. So as Derek mentioned, another focus of our group is drug repurposing. So I had a mid-career fellowship from the Advanced Queensland program, the Queensland government, wherein we were working on evaluating drugs which are approved for other conditions, which have the potential for being repurposed or repositioned, as we call it, for MND or Parkinson's or brain diseases more broadly. So one of the reasons for taking a drug repurposing approach, as you'd see with some of the drugs that we're working on, is that there's a shorter time scale, and particularly with complex brain diseases where inflammation is a common thread across several diseases, including cancer and diseases like arthritis, where a lot of progress has been made in terms of finding out the pathways and developing drugs over the last 20 years. So our hope was, by understanding or repurposing some of those drugs, or finding the pathways that are important between which of those inflammatory pathways are important in Parkinson's or MND, we could then repurpose some of this effort or billions of dollars that are spent developing these drugs for other inflammatory diseases and find out which ones could have potential for brain diseases like Parkinson's and MND. So we've just got started with MND this year, so I don't have a huge amount of work or data to present as Derek had for their incredible metabolism program. So we've started this work in collaboration with Derek and Shu and others at Rob Henderson and Pam McComb at the Royal Brisbane Hospital. And I'll present some of the data towards the end, but some of what I wanted to cover initially was what is the evidence for neuroinflammation in MND pathology in clinical studies and animal studies, and then also what are the emerging sort of concepts and therapeutic avenues by which we could target inflammation as a therapeutic strategy for slowing or halting disease progression in MND. So if we look at the clinical evidence for inflammation in MND, so it's been known for probably a decade or so now that there's elevated inflammation markers present in the blood and body fluids of MND patients. So if, for example, we find elevated interleukins, IL-1 beta, other inflammatory markers. So there's also highly activated immune cells that are evident in the brain and spinal cord of MND patients. So postmortem sections, for example, or even in live patients now, we could use MRI and PET imaging to image inflammatory activity in the brain and spinal cord. So there's also infiltration of immune cells. So the peripheral immune cells from the blood do infiltrate into the brain and spinal cord. So there's evidence from this from postmortem patients again, and also from animal studies. And then finally, there is some epidemiological evidence linking long-term use of anti-inflammatory drugs. Those are things like aspirin and other anti-inflammatory drugs that you can get over the counter. And also prescription anti-inflammatory drugs. There are some studies linking long-term use of these drugs with a slightly lower risk of MND. But with a lot of things in the scientific literature, some of this is still a bit controversial, and some of it hasn't been confirmed by other future studies. But there's a few studies that have shown this, and this is the case with a lot of neurodegenerative diseases, not just MND. Even Parkinson's has these epidemiological links. All of them haven't translated in the clinic, for example. So there's an epidemiological link for Parkinson's and aspirin use, for example, similar to what's been shown previously or suggested for MND. But that strategy hasn't always worked out in robust clinical trials. So if you see here, this is just an image of some inflammation markers from the inflammasome. So the inflammasome's a complex that's present in immune cells. It's sort of the master regulator of the inflammatory response in most brain diseases. So if you look here in ALS patients, you have increased elevated inflammatory markers and inflammasome markers here. So in this case, NLRP3 is what we use to measure the inflammasome. So NLRP3, there is a lot more NLRP3 activation here in green. And most of that is on these inflammatory or activated immune cells in ALS patient brains. So similar to that, you've got other markers here, ASC, which is an adaptive protein for the inflammasome complex, and markers like IL-18 and interleukin. So all of these markers here are elevated in sporadic ALS patients and in patient brains. So, and there's a lot of evidence from animal models, knockout mice and things, where these proteins have been shown to propagate inflammation in the brain, the spinal cord, and lead to neurodegeneration. And then that drives the disease progression. So imaging of inflammation in MND patients is something that's been increasingly studied from the context of earlier diagnosis and also for disease monitoring to see if we can develop probes that could be used in clinical trials, because inflammation occurs in varying degrees in different people, but it's a common thread amongst all forms of MND and ALS. Inflammation, there are varying degrees of inflammation that are evident. So the idea here is that if you could have an imaging agent, a sensitive imaging agent, you could use it or develop it for not just diagnosis, but also monitoring the efficacy or the effectiveness of clinical trials. So there's several of these probes available, the TSPO compounds, but all of them essentially, they work by binding specific immune receptors that are expressed on these immune cells, which are involved in inflammation in the brain and spinal cord. So you can see from this probe that was developed at MassGen, PBR28, the red and yellow in basically the heat map here suggests increased inflammation in living ALS patients. So unlike postmortem studies, which were on the previous slide, these studies have confirmed that inflammation is evident. It's an ongoing process. It starts very early in the disease, and it continues all the way until death. And you can see even stronger signals for inflammation in postmortem patients compared to the living patients that we've observed here. So if you come to the question of how inflammation can drive neuronal death, that's been something that we've been working to figure out. across the board. So there's several different theories, several pathways that have been put forward. All of them ultimately have been, most of these studies have come from animal models. So the premise of all of this is that blocking pathways of inflammation is protective in several different animal models of MND. So there have been drugs that have been used to probe this. There have been knockout mice for different pathways, and whether it's a SOD mouse or a TDP43 mouse, most of these pathways, most of these therapeutic strategies have been based on dosing these mice and looking at survival and measuring immune and inflammatory markers in the brain. So the caveat here is that animal studies so far across the board, even though these drugs have worked well, the knockouts seem to have increased survival for these inflammatory pathways. Most of these studies have not yet translated well into effective treatments. And most of us who work in this field are acutely aware of this. We still don't have enough of an understanding of the underlying mechanisms that are involved in inflammation. So, and basically that is where most of the research needs to be done. So there are several pathways that have been implicated in ALS. And so NOX2, TNF-alpha, alpha-beta, the interleukins, there's been oxidative stress, mitochondrial dysfunction. It becomes really complicated very quickly. So essentially the underlying mechanisms are quite complex, but there's this concept emerging of protective immune cells versus pathogenic or harmful immune cells, or protective inflammation versus harmful inflammation. So you have what's called a protective inflammation resolving sort of phenotype in cells, the M2 phenotype in microglial cells. So these M2 microglia or M2 macrophages are found in healthy individuals. And this balance, and in neurodegenerative diseases, you see an pro-inflammatory or pathogenic immune signature emerging. So that's the M1 phenotype. So in the healthy individuals, or even before the onset of ALS, for example, the prevailing theory, the prevailing hypothesis is that these immune cells have a protective sort of surveillance phenotype. They're called surveillant microglia, and they mostly function as housekeeping cells to protect the brain and spinal cord from inflammation. And somewhere along the way, there's a switch, possibly over a period of decades, years, to unresolving chronic phenotype where these cells take on or acquire a pathogenic or harmful inflammatory phenotype. So that is essentially where the switch occurs. That's an area that's being highly studied as to why inflammation doesn't resolve, what makes these cells go from a healthy state into a constant state of activated inflammation in the brain and spinal cord. So that's a theme across the board that we don't yet understand, that just the research just isn't there yet. So, and then what propagates this inflammation, or what keeps these cells in this constantly activated inflammatory state? So we have a few answers in that space. So because there's an accumulation of protein aggregates in the brain and spinal cord, we have those protein aggregates have been shown to be a trigger. And because those protein aggregates are not clear, they accumulate and persist and spread even. That's been linked to what could be maintaining these cells in that inflammatory state. So there are some answers in that space. So, and one of the more exciting avenues of research that's coming out there is if you're able to switch these cells, these immune cells, back to a healthy or protective phenotype, the M2 phenotype, there's some evidence from Parkinson's and Alzheimer's disease, at least, wherein these cells are able to clear some of these protein aggregates and that could prolong the survival of these neurons and sort of put a halt to the disease progression. So that's all been only shown in mice so far. We've seen that in some of our models of Parkinson's, and it's been demonstrated in Alzheimer's. I'm not aware of that being demonstrated yet in ALS, but that's a concept that we're working on, and several others are also exploring in this space. So another emerging area in inflammation and neuroinflammation in particular, and is the role of the gut microbiome, and that gut microbes can tune inflammation and lifespan in models of ALS. So this really exciting study was from Harvard and Mass Gen. So they had shown that the same mice, the C9orf72, if they were housed in one facility, they found that they were almost completely protected from, they had longer survival and much less severe disease. And the only difference in both of those mice was the housing facility. So the animal facility in which they were housed. So a lot of us who work with inflammation, and anecdotally, we've sort of, it's something that's quite common to us. We've had this in all of our previous models with Parkinson's. We know some of them work better in one animal facility, and whereas if depending on how clean or dirty it is. And over the last decade or so, we've sort of understood why that is. Historically, that wasn't very clear. So now we know that the gut microbiome is different in different animal facilities, depending on how clean or dirty it is. And basically, that could have a profound impact on the inflammatory response. So this is an area we're actively studying in Parkinson's, and we're hoping to get into for ALS as well. But it's an emerging area that sort of, it joins a lot of the dots. It answers a lot of the questions that we've had in terms of neuroinflammation as to how it could start and how it could be propagated and how inflammation is sustained in all of these diseases. So basically what they found here is that in the Harvard labs, the C9orf knockout mice succumbed to the normal inflammatory phenotype that they had. And then when they were housed at the Broad Institute, these mice lived normal lifespans despite having some inflammation that you normally get in these models. So the team went through some fantastic detective work, and eventually they found out that the difference in the gut microbiomes explained this variance. And so this has led to a lot of different theories as to which microbes are involved, how these just a change in the microbiome in genetically identical mice of mouse models of ALS could have such a profound impact. So, and there's been other studies that have followed up on this, or in parallel have sort of made a very strong case for a link between microbiome and microbiome changes. So this paper earlier this year in Disease Models and Mechanisms, so they used the SORD model, the SORD1 mouse model of MND. And so they went through characterizing. What they were interested in understanding is how the microbiome dysbiosis, which it's quite well known that occurs in this model, how that fits in terms of the timeline with inflammation, with the common disease phenotypes, so muscle atrophy and things like that. So what they did was they went into great detail and characterized microbiome changes, inflammatory markers, and epigenetic markers, and the disease phenotype at different disease stages. And they sort of broke that down. And what they found here again was that dysbiosis occurs quite early. So this is similar to it parallels what we see in our Parkinson's disease studies with the mice that we work with. We found a very strong change in the microbiome of these mice way before you get any changes in inflammation. So before we started studying the microbiome, the inflammation was the earliest signal that we could find in most of these disease models of neuroinflammation and neurodegeneration. So we're increasingly appreciating now that the changes in the microbiome even precede the early changes of what we thought were early changes in inflammation. So, and of course, most of these changes in inflammation and the microbiome seem to precede the motor symptoms or the neurodegeneration that occurs. So there's a lot of evidence pointing to the microbiome changes occurring first and then inflammation happening on the back of that. And that somehow that the microbiome changes can sustain the harmful microbiome changes that occur, or the gut dysfunction that occurs can sustain this inflammatory process. And that eventually leads to the loss of motor neurons or neurodegeneration. So in terms of how does inflammation drive motor neuron death? So what we know, there's more that we don't know here than what we actually know. So we do know now that inflammation starts very early in the disease process. Inflammation doesn't appear to resolve in MND. So healthy inflammation is usually self-resolving. So healthy inflammation, if you have an infection or an injury, for example, so healthy inflammation kicks in, it's intended to quickly fix the infection, the infecting bacteria, the virus, whatever it may be. And then it resolves. It's usually self-resolving. But what happens in most brain diseases, including MND, is that we don't understand why this inflammation is unresolving, what sustains it. We've got some clues now from the microbiome as potentially one of the changes in the microbiome and the metabolic changes that happen on the back of that, underlying or underpinning this sustained inflammation that we see. There's still not a lot of direct evidence for that, but there's a lot of pieces of the puzzle that are pointing towards that at the moment. So, and then chronic inflammation is, of course, detrimental to neuron survival. So one of the most highly cited papers in review articles for neuroinflammation, and which first proposed this concept of cyclic neurotoxicity from Block et al. In 2007. I believe this is probably the most cited review article in neuroinflammation. And this paradigm proposed back in 2007, controversial at the time, has now become dogma, wherein you have microglial activation, you could have inflammatory triggers, environmental or other triggers, which produces neurotoxic factors that drive neuronal damage. There's still a lot of evidence that environmental factors, pesticide exposures, chemical exposures can also trigger directly damage neurons, and that leads to some triggers of inflammation in these immune cells. But basically, at the end of the day, it's unclear where this process starts. But what's clear is that there is a cycle of self-perpetuating neurotoxicity that occurs, wherein the inflammation drives neuronal death, and this neuronal death and the accumulation of the protein aggregates sustains the immune cell inflammation and infiltration of these immune cells into the brain and spinal cord. So that's basically where the therapeutic strategy comes from. If you can block this inflammation, you can prevent neuron loss, but you can also, if you can resolve this inflammation, that gives you another avenue by which we could treat or shut down the progressive loss of these motor neurons. So there are increasingly a lot of trials targeting inflammation in ALS. So there's enough evidence accumulated in animal models for many mechanisms to now allow for this. So most treatments are based on the premise that inflammatory mechanisms promote neuron death in MND. So there are a few immune modulators. So this is from a review earlier this year. So this is a monoclonal antibody that can normalize inflammation in sporadic ALS patients. Ibutylast is based on reduction of brain atrophy in multiple sclerosis, but they've shown that these mechanisms are relevant to MND and ALS as well. So there are trials in phase 2 and phase 3 for most of these drugs. And then as part of the Healy platform trial, Zalucoplin and Bediperstat are immunomodulators that are being tested. So this platform is quite unique, wherein you have inflammation and other mechanisms that are being tested together in sort of a linked clinical trial, wherein you have one placebo group and then different drugs sort of in combination. So it's a platform trial and it's unique in that sense. So inflammation is being tested along with other neuroprotective mechanisms. So, and then sodium phenylbutyrate is another drug that was just published, a clinical trial that was shown to have a small effect on survival of people with ALS. So this was in the New England Journal of Medicine earlier this year. So it had 137 patients with ALS, and 89 of them were on the drug, or 48 were on placebo. And with the patients on sodium phenylbutyrate, the combination, they had a slight increase in survival and outcomes, although the secondary outcomes like muscle strength and vital capacity were not really different. So one of the interesting things that we know about phenylbutyrate from most of our studies in Parkinson's and inflammation more broadly, neuroinflammation more broadly, is that it's been shown sodium phenylbutyrate in the Parkinson's disease world, basically. It's been known from the early 2000s that this compound, this pathway, has a profound impact on inflammation. I don't think the study went into a lot of inflammatory endpoints, but the fact that it's one of the emerging drugs that seems to have an effect in ALS, which is still, I think, needs to be repeated in larger patient populations. But basically, it was in the New York Times earlier this year as well. And basically, this drug does have profound impacts on the inflammatory response and the immune response. So it's likely that there is a component of inflammation that this drug is targeting, or this combination of drugs are targeting as well. So moving on to some of the work that we've been doing with in MND using repurposed drugs. So we were fortunate, like I said, to get some funding from MNDRA this year to look at one of the pathways that we found we've been studying in Parkinson's disease, but in collaboration with Shu and Derek and our clinical collaborators, Pam and Rob Henderson at the Royal Brisbane, we were able to find that this pathway called Bruton's tyrosine kinase is involved in, is strongly activated in the spinal cord of our MND mice. So we use a SOD G93A, a very common model of MND. So the cool thing about BTK is that this particular drug is involved in leukemia. So it's involved in immune cells. So B cells are a specific population of immune cells that are highly activated in several blood cancers, and they become oncogenic. So there are FDA-approved drugs targeting this pathway that have shown to be effective in people with MND, with blood cancer. So what we are studying essentially with this, so we're taking a drug repurposing approach, and the reason for us taking this drug repurposing approach is that there's a shorter time scale. So if you start with a new drug for this pathway, you can spend 12 to 16 years, $1 to $2 billion, and still not get very far in terms of clinical efficacy. So what we're doing is trying to take a shortcut in that space by using drug repositioning. So basically, a shorter time scale, because most of these drugs have already passed to drug discovery, preclinical testing in different phases for other diseases, and they've been approved. We decided to use or test this blood cancer drug that's FDA approved in our SOD spinal cord SOD mice to see if it blocks inflammation and if it can improve survival. So we were fortunate to get a grant for this, and we've been testing this drug since around the middle of this year. A lot of our studies were disrupted by COVID, but we've just completed our first set of drug studies. We don't yet have a lot of the data analyzed yet, and we're still continuing treating our mice over the Christmas break and early next year. So, but our initial results are quite promising. We've found that the FDA-approved inhibitor drug, the cancer drug, definitely works really well in immune cells, isolated microglia. So these are microglia for which we use ATP to activate the inflammasome. You see here that there is a strong inflammatory response with ATP and LPS, which is we normally use to prime the inflammasome. And with the BTK inhibitor, the FDA-approved drug, we could see a pretty good reduction. So our initial studies from the SOD mice is we are seeing that these mice which were on this cancer drug are able to maintain their body weight. And we've recently just euthanized a lot of this, completed the study, and we're looking at first at the inflammatory markers in the brain and the spinal cord. And with our ongoing studies, we have a student and a postdoc looking at if this drug can improve survival. So what this essentially allows us to demonstrate is proof of concept, so that if we have efficacy in the SOD model, it can take us one step closer towards translating this drug, this therapeutic approach towards the clinic. So We also started working with a company that makes this drug. And so if our results are promising, and we think they are so far, hopefully next year sometime with some additional funding for a clinical trial or a small clinical study, we'd be able to take this directly into the clinic because it is FDA approved. So in terms of the unresolved questions, they're pretty much the same in Parkinson's, Alzheimer's, or even MND when it comes to neuroinflammation. So what are the triggers in MND and where do they start? Why is inflammation unresolving? Can this be shut down using drugs, which is what we're sort of looking at and a lot of others are looking at in terms of a therapeutic approach? How does gut microbiome link into ongoing inflammatory responses? And can immune cells be harnessed to clear protein aggregates in MND? And can immune inflammation signatures be harnessed for earlier diagnosis of MND? So we're very grateful for our funding from MNDRA and our collaborators at UQ and the Royal Brisbane. And we also have a very hardworking team of people who work on Parkinson's, and the ones in blue here are the ones who work on our MND project. Thank you very much.
Great. Thank you, Richard and Derek, for a fantastic comprehensive overview of two kind of growing areas in MND research. So we've got time for a few questions here. So we've got a couple of questions coming from the audience. So the first question we have is for Derek. And this is, can you affect your metabolism by altering your levels of activity? I'm guessing physical activity. So can you mitigate some of these changes by exercising more or less?
Okay, so I guess outside of MND, if we want to increase our metabolism, we would exercise, build muscle, and that would help us actually increase our metabolism. I'm not saying that's not a good idea. In fact, I think it's a really good idea to do exercise up to your level that you can exercise in MND. There's good evidence to suggest that it's beneficial and helpful, but of course not pushing yourself beyond your own limits. But I don't think that exercise is actually going to address hypermetabolism. And the reason I say it is because we actually don't know what hypermetabolism is coming from. Like I said, I mentioned some of the muscle, but really it's also possible that hypermetabolism would be associated with things like inflammation. It could be even the genetics of the individual, and it may also be things like changes in the neuron. So I'm sorry to say, I don't know if exercise is going to improve metabolism in terms of reversing hypermetabolism.
Okay, thank you. And we have a question here for Richard. Are there, is there any evidence that, say, levels of more generic, I guess, inflammatory markers such as ESR, do they mirror the, or do they act as biomarkers of neuroinflammation?
Sorry, what marker was that, please?
ESR is a circulating marker of inflammation.
Okay.
I can't remember exactly what it stands for.
Yeah, so I think, like Derek mentioned, there is a lot of variability so far that we've observed, even in the immune response. If you look at the variability in the published papers, not everybody seems to have some level of inflammation, but you have people who have it's a spectrum. So it's a lot of patients have some markers that are elevated, some others don't. Overall, so far, I don't think we've lined up a specific marker like ESR that could be definitively used as a biomarker.
And is there any evidence that very powerful anti-inflammatory drugs such as the anti-TNFs or some of those other biologics have any effect in MND? I know they've been trialed in dementia and I don't think they work there. Do you know if anyone's had a look at them?
Some of the ones that I mentioned in my talk, they are antibodies, but I think we'll have to see, because a lot of these drugs have shown, like the TNF that you mentioned, they have very promising results in dementia patients. And even actually there was one patient who almost had a temporary reversal of the symptoms that's been documented with the TNF antibodies. But then when they tried these in a larger cohort of patients with diseases at different stages, the drugs weren't as effective. So I think that's part of the problem that we face, that everyone's at different stages and all the drugs may not work at, the pathway might only be most effective, blocking the pathway might be effective in, say, earlier patients at an earlier disease course, but not when they're in the middle of the disease, for example, or mid-stage disease.
And another question for Derek. You talked about evidence of hypermetabolism being related to survival and disease severity. Is there any evidence as to whether your levels of metabolism affect your propensity, susceptibility to MND? Are people either, say, people who are skinnier or larger, is there any evidence that you may be more or less likely to develop MND, or along those lines? Has any work been done there?
There's been a lot of work done there, and I think that's a really interesting question because so when we talk about people being skinny, and we really refer to things like BMI. BMI is that thing called body mass index, and there's a lot of evidence out there to suggest that people who are really skinny and really lean are more likely to get MND. Those individuals that are actually overweight towards obese are less likely to get MND, and even if people with a high BMI do get MND, they get it later in life. And so it goes pretty much against this notion that we have that being overweight is bad for you. In MND, it seems that overweight can protect you from getting MND, and if you do get MND, you get it a little bit later in life as well. It's a bit of an anomaly that we see in the health system.
And I'm not sure if this is a useful question or not, but if you are unfortunate enough to be diagnosed with MND, and perhaps it's fairly early in the disease course, is there any benefit to try and put on weight?
I think that's a really good question. That's a fantastic question. So first of all, I think gaining weight and increasing your body weight is not going to be good for everyone. So depending on the type of MND you have, putting on that extra weight might actually make life a little bit more difficult. But a really nice study was published just earlier this year, actually looked at high calorie feeding, and particularly eating a fatty rich diet as a way of slowing down the progression of MND. And in that study, they actually found that those people who are maintaining their body weight and those people who are adding fat to their diets, who had a more rapid progressing type of MND, were able to slow the course of their disease. And so there is some evidence to suggest that by putting on some weight, especially if you have a rapid progressing disease, and by making sure you don't lose that weight, I think that's the important one, you can have a big impact on the course of disease. And so I think the message from that was that it's really important to have a good, close working relationship with your dietitian, and to try and make sure that you stay on top of diet and healthy nutrition very early on.
Okay. So there's a couple of more generic questions here from Bruce Bird, which you may both want to have a go at. So yeah, he says he's very impressed with the presentations you've had over the last few weeks, but he's seeing a lot of tests done on humans and mouse models, but just wondering how much more research and time is it going to be before we start seeing some new drugs other than Riluzole?
Richard, did you want me to go first? Do you want to take that one on first?
Yes, please. I think you're more qualified to answer that as an MND expert.
I really don't think that any of us are qualified to answer that question. And the reason for that is, first of all, apologies for the confusion out there. There's a lot of research going on, and it can be very confusing. But perhaps apology is a bit of the wrong use of words here, because it's kind of a testament of just how much energy and effort the research community is putting into MND research at the moment. I have not really seen this level of interest in MND since I've started doing MND research more than 10 years ago now. So it is unfortunately a consequence of just the sheer volume of energy that's being put in there. In terms of finding a treatment and a cure for MND, I don't think that we are close to finding a cure for MND, but I certainly think that we are getting closer and closer, discovering real therapies that's going to have a big impact on individuals. We've got more clinical trials running for MND now than we've ever had before. More drugs are being tested, and a lot more money is being invested into the research. And I think that's going to be the key. In terms of the global effort, Australia is doing exceptionally well when it comes to MND research, because we have organizations like the MNDRA supporting that research. And I think that the more we support that research, the better we'll do. And I don't know of a single researcher in Australia that doesn't work with an international group or that doesn't collaborate widely. In fact, if you were at the MND meeting last year in Perth, Australians were the third most prevalent group of researchers there. And so I think that Australians are working exceptionally well with international teams and are working exceptionally well overall to actually conduct research, but it's not enough. We really have to do more. And hopefully we will see more research happening very soon.
Yeah, and I'd like to add a couple of takes to that. One on the diversity of drugs. I mean, there are, as Derek said, a lot of drugs coming through in phase 2 and 3 at the moment, trials, but because MND is such a heterogeneous disease, I don't think there's ever going to be one cure-all drug. And I think we need to keep producing more and more different approaches. So every patient is hopefully in the next few years will have a personalized medicine approach. We'll look at the symptoms and we'll have developed a lot of different treatment approaches. So you can basically have a cocktail of treatments that will suit your specific form. And that's why we need to keep producing more and more research into models, and it has to be tested thoroughly before it can come through into clinical trials. So we have to keep that pipeline full, I think, at the moment.
Ketan, you mind if I just quickly add something that I actually didn't mention? And we've really been focusing on drugs here, but the other game changer for MND has been just the level of knowledge that the clinics are gaining and the strength from the clinicians in developing these multidisciplinary teams. I mentioned how there are different factors that impact the rate of progression for people. Well, one of the most powerful things has been the introduction of that multidisciplinary clinic. And that certainly has had a huge benefit. And that's why today, if you get diagnosed with MND, you will survive longer and you will have a better outcome compared to somebody diagnosed 25 years ago.
Yes. And a last little additive, as Derek was saying, I think in Australia, we probably bat way above our weight as such, because even though we're a smaller country with probably smaller resources, our discoveries are really great. We've got drugs that have come out of Australia that are currently being tested. And many of our researchers here are really considered top of the game across the world, are really well-respected international researchers. So I think your dollars invested in research in Australia are probably better spent here than anywhere else in the world for the outcomes we're getting. So we're running a bit late over time, so we'll have to bring it to a close. I was one quick question. This is probably for you, Richard. The differences in between the different mouse models between labs, how much is that going to have implications for study replication and confound some of the interpretation of these results?
Yeah, look, I think that's a very important point, because replication is something like I was saying, we've seen this, observed this before in our Parkinson's models, as that's the difference in between animal houses, the clean versus dirty facilities. So, but I think in that particular study that I mentioned, they went to great extents to replicate their studies and to confirm which microbes, down to which microbial populations were involved in that effect. So that they actually made an entire study out of that. But increasingly, that's become something that we've become more and more aware of. Certainly in our Parkinson's work that's funded by the Fox Foundation, we are now putting into our budgets microbiome sequencing for all of our models, so that we actually know what the microbiome is before we start the study and the end of the study. So that's sort of becoming best practice. And I think that'll improve reproducibility quite a bit. There's been papers published about that as to best practices for the microbiome studies. And I think those not yet, they are standard practice in the microbiome world with microbiologists who study this for, say, inflammatory bowel disease or other diseases of the gut. But I think that should be used more widely by almost everyone studying even neurodegenerative diseases. And I think that'll change replication, or that'll improve replication quite a bit.
Great, thank you, Richard.
So I just quickly add one very quick small thing to that.
Please, Derek.
I do want to caution people when it comes to interpreting the mouse studies. Mouse studies are fantastic for identifying single pathogens and multiple pathogens and how they impact. But remember that as people, we don't live in those environments. And so our microbiome is very complex. And so I think running alongside those mouse studies, we really need to start investing in very large cohort studies looking at the microbiome in the real world as well. Because it's only when we combine those two studies that we can really tease this one apart. Great.
Thank you, Derek. So I'd just like to finish up by thanking both Richard and Derek again for taking us through some fairly complex areas of research. Fantastic overview, both of you. And probably just as important, if not more important, I'd like to thank the audience for coming along and making and hopefully listening and enjoying these talks. please feel free. You'll be receiving recordings either through Zoom or Facebook. To please feel distribute that through your networks so more people can hear about this fantastic research. We'd welcome your feedback. This is the first time we've run such a series. So please write to us on Facebook, Twitter, directly onto our through email, and tell us what you thought of the series, any ideas for improvements, what topics you'd like to hear about, because even though we'll probably run MND Connect hopefully next year, COVID permitting, we'll still think about running another series like this again as well, because the more outreach we can get, the better. We would like to make sure everyone can hear about our research. So all it remains is to thank everyone involved, and hopefully everyone can have as enjoyable a break as they can. I think after this year, everyone deserves a decent break. So please enjoy your Christmas and New Year. So good night, everyone.
Good night, everyone.
Thank you.